Welcome to Creative Biolabs. Microfluidics is a technology for precise control and manipulation of microscale fluids, especially sub-micron structures. It's also known as lab-on-a-chip or microfluidic chip technology. It integrates the basic operation units such as sample preparation, reaction, separation, and detection in the biological, chemical, and medical analysis process into a micron-scale chip to automatically complete the entire analysis process. We will give a brief introduction to microfluidics from the following four aspects: history, introduction, characteristics, and applications. The first aspect is the development history of microfluidics. In 1959, Professor Richard Feynman, winner of the Nobel Prize in Physics, proposed the microelectromechanical system, or MEMS. The basic concept is to use semiconductor technology to miniaturize the mechanical system in real life to form a microelectromechanical system. In 1990, Manstadol first proposed the concept of miniaturized total analysis system, or micro-TAS. In 1995, Caliper Life Sciences, the first company engaged in microfluidic chip technology, was established. In 2003, the microfluidic technology was named by Forbes magazine as one of the 15 most important inventions affecting the future of mankind. In July 2006, Nature magazine published a special edition entitled Lab-on-a-Chip, expounding the research history, current status, and application prospects of lab-on-a-chip, and pointed out in the editorial comments that the lab-on-a-chip may become technology of this century. So far, the strategic significance shown by the lab-on-a-chip has been recognized by the academic and industrial circles at a higher level and on a larger scale. The second part is a brief introduction to microfluidic technology. This introduction will be mainly divided into the following three major aspects. We will first outline the microfluidic technology. The second aspect is the principle of microfluidic technology. And the third is the components of microfluidic technology. Microfluidic chip is also known as lab-on-a-chip or biochip. It uses MEMS technology to microfilm a large laboratory system on a glass or plastic substrate, thereby replicating the entire process of complicated biological and chemical reactions, and completing experiments quickly and automatically. It is characterized by constructing fluid-containing channels, reaction chambers, and other functional components on a micrometer scale, and manipulating the movement of micrometer-sized fluids in tiny spaces, thereby constructing a complete chemical or biological laboratory. Microfluidic chips use semiconductor-like microelectromechanical processing technology to build a microfluidic system on the chip. The experiment and analysis process are reprinted on the chip structure, composed of interconnected paths and liquid phase chambers. After the biological sample and reaction solution are loaded, methods such as micromechanical pumps, electrohydraulic pumps, and electro-osmotic flow are used to drive the flow of the buffer in the chip to form a microflow path, so that one or more continuous reactions can be performed on the chip. Various detection systems, such as laser-induced fluorescence, electrochemistry, and chemistry, as well as many detection methods combined with analysis methods, such as mass spectrometry, have been used in microfluidic chips. Fast, accurate, and high-throughput sample analysis has been realized. A complete microfluidic system includes many components, mainly micropumps, microchips, microvalves, and detection units. Among them, as the heart of the microfluidic system. The micropump is the power source for microfluidic transportation. These pumps have been mainly categorized into pressure pumps, piezoelectric pumps, peristaltic pumps, and syringe pumps. Microchips can also be classified according to different dimensions. And according to the material of the chip, it can be divided into silicon, glass, organic polymer materials like PMMA, and paper materials. Based on the types of channel array, there are single-channel and multi-channel chips. According to the network shape, it can be divided into linear, spiral, curved snake, polygon, and folded. In the third part of this video, we will discuss the characteristics of microfluidic technology, that is, the advantages and disadvantages. The main advantages of microfluidic technology are as follows. 1. The integration of miniaturization and automation. Microfluidic technology can combine multiple steps of sample detection on a small chip, miniaturizing and automating the entire detection integration. 2. High throughput. Since microfluidics can be designed as multiple channels, multiple items can be tested in parallel on the same sample as needed. Thereby, the detection time is greatly shortened, the detection efficiency is improved, and high throughput detection is realized. The third advantage is the low consumption of detection reagents. Due to the miniaturization of integrated detection, the reaction unit cavity on the microfluidic chip is very small. The reagent usage is far lower than that of conventional methods, and thus the consumption of reagents is greatly reduced. 4. The demand for sample size is small. Since the test is only completed on a small chip, the sample size required to be tested is very small, of which microliters or even nanoscale could be enough. And 5. Less contamination. Due to the integrated function of the microfluidic chip, all the operations that were originally required to be completed manually in the laboratory are integrated into the chip and can be automatically completed, so that sample contamination during manual operation is reduced to a minimum. With the above important advantages, microfluidic technology has become the first choice for POCT. There are 4 main disadvantages of microfluidic technology. First, the core technology lacks specifications and standards. In the industrialization of microfluidics, due to immature technology and the lack of corresponding standardization, it is currently impossible to realize the universalization of components. The second disadvantage is a serious shortage of relevant talents. There is a serious shortage of multidisciplinary talents, corporate R&D personnel, and specialized market personnel. And third, the current production cost is high. For the microfluidic immunoassay chip, since the analysis chip is just for one-time use, the detection cost is increased. The fourth is the difficulty of the technology platform. For example, problems such as the immobilization of antibodies and the sealing of the surface of microchannels still exist and should be improved. The fourth part is the application of microfluidic technology. Creative Biolabs can provide professional microfluidic-related services. It can be mainly applied to the following 4 aspects. Biological analysis based on microfluidics, including microfluidic-based analysis in cell mode and microfluidic-based exosome separation analysis. Clinical diagnostic analysis based on microfluidics, including pathogen detection and analysis based on microfluidics, cancer detection and analysis based on microfluidics, etc. Food safety analysis based on microfluidics, including the detection of pesticide residuals, pathogenic bacteria, heavy metals, and food additives. Environmental monitoring and analysis based on microfluidics, Detection methods include electrochemical detection, optical detection, MS, and etc. Creative Biolabs is a leading customer service provider in the field of cancer next-generation sequencing, with a professional platform and rich experience. More information about Creative Biolabs can be reached by visiting www.creative-biolabs.com or by calling 1-631-871-5806. Or you can send an email to info@creative-biolabs.com.